Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast covers all things adenosine triphosphate, or ATP. I'll talk about where and how ATP is produced. I'll talk about the structure of ATP. Then I'll get into some of the thermodynamics of ATP and talk about how it's paired with biological reactions in order to drive them. And then lastly, I'll talk about a few specific cases that I've seen on the MCAT regarding ATP. That's going to be the sodium-potassium pump, or the sodium-potassium ATPase. I'll talk a bit about protein phosphorylation. And then lastly, I'll talk about ATP binding cassette transporter protein, or ABC transporter proteins. I know this kind of seems like a focused or kind of an obscure topic, but I have seen ATP come up quite a bit on the MCAT, or at least topics regarding ATP. So I think it's fairly high yield. And this material will show up on 2 of the 4 MCAT sections. That's the biochem section and also the chem physics section. With that said, I hope you find this podcast helpful. I want to start this podcast by going over the structure of ATP. Adenosine triphosphate is a nucleotide, and remember, the definition of a nucleotide is a monomer that's composed of a nitrogenous base, a 5-carbon sugar, and then 1 or more phosphate groups. So ATP in specific has the purine base adenine, which is the A in DNA. And I don't mean like the letter A in the word DNA, it's the actual A's that make up our genome. And I think it's important you understand the difference between a nucleobase, a nucleoside, and a nucleotide. And I think I went over this in the Biomolecules podcast, but I'm just going to quickly review because I think this is something that's important to understand for the MCAT. So a nucleobase is a nitrogenous base that's incorporated into nucleotides and nucleosides. And this is adenine, guanine, cytosine, thymine, and uracil. And these are the A, G, C, and T, and also U that show up in DNA and RNA. A nucleoside simply consists of a nitrogenous base, also called a nucleobase, and a 5-carbon sugar that's either ribose in the case of RNA or deoxyribose in the case of DNA. And these nucleosides are named a bit different than the nucleobases. So the ribonucleosides are adenosine, guanosine, uridine, and cytidine, while the deoxyribonucleosides are deoxyadenosine, deoxyguanosine, thymidine, and deoxycytidine. So take the nucleobase adenine and add on a ribose sugar with 2 hydroxyl groups on it. That's going to now be adenosine, and if I was to remove one of the hydroxyl groups, it would be deoxyadenosine. So that's a nucleobase and a nucleoside. A nucleotide is composed of a nitrogenous base, a 5-carbon sugar, which is either, again, ribose or deoxyribose, and then 1 or more phosphate groups. So let's take, again, adenine, which is a nucleobase. You add on a 5-carbon sugar, say ribose, that becomes adenosine. Now you add on a phosphate that becomes adenosine monophosphate. In the case of ATP, our friend, we know that that's adenosine triphosphate. So that's an adenine nitrogenous base with a ribose sugar and with 3 phosphate groups. So that's kind of the overall structure of ATP. And, it's pretty easy if you just remember how to name these nucleobases, nucleotides, nucleosides, how you can kind of think about the structure once you hear adenosine triphosphate, right? And the last thing here I want to talk about in terms of the structure of ATP is the naming of these different phosphate groups on the ATP. So there's 3 phosphate groups, and there's a phosphate group that is attached to the ribose sugar, which is closest to the purine base, the nitrogenous base. Then there is a phosphate group that is in between both the other phosphate groups. Then there's a phosphate group that is on the outside of the other phosphate groups, kind of furthest away from the ribose sugar and the nitrogenous base. These different phosphate groups are denoted using different Greek letters. So the phosphate group that's closest to the ribose sugar and actually attached to the ribose sugar is the alpha phosphate group. Then the middle phosphate group is the beta phosphate group. And then the outermost phosphate group, kind of on the end, is the gamma phosphate group. And typically when ATP is reacting with some other substrate and it's losing a phosphate and turning into ADP, you're losing that gamma phosphate, which is the phosphate on the end. And ATP is a relatively unstable molecule. You have these three negatively charged phosphate groups all packed pretty tightly next to one another. And so I'll get into more of this in the thermodynamics part of this podcast, but just understand that you have these three phosphate groups that are all packed together, and you have the gamma phosphate furthest away from the nitrogenous base. Then you have the next phosphate is the beta, and then the next phosphate is the alpha phosphate, and so you have gamma, beta, alpha, shout out fraternities, and that alpha phosphate is attached to a ribose sugar, which is then attached to an adenine nitrogenous base, and that's ATP. In the next segment of this podcast, I want to talk about ATP production. So ATP is produced in numerous catabolic reactions, which are just reactions that break down large organic molecules. And in terms of production of ATP, I can kind of talk about this in two different ways, right? So I can either regenerate ATP from ADP by just adding on a phosphate group, and that's a lot of the metabolic processes that we usually talk about. Or I could talk about biosynthesis of ATP molecules, where I'm talking about actually creating these nucleotides from scratch. And I will say that these two different categories of ATP production aren't separate, right? They're not just standalones. My understanding is that we can synthesize de novo AMP or adenosine monophosphate. And then what we do with that monophosphate is then we add on multiple other phosphates to either make ADP or ATP. In other words, there is a nucleotide biosynthesis pathway, which the nucleotide in this case is adenosine. And what that pathway is going to do is it's going to take the adenine base, it's going to take the ribose sugar, and it's going to take a single phosphate, and it's going to all push those all together, and it's going to create an AMP molecule. And then from there, you can feed it into what I'm calling the regeneration pathways that just add phosphates to existing AMP and ADP to make ADP and ATP, respectively. So I want to start by talking about that second category of production of ATP that I talked about, which is regenerating ATP from ADP. And there's three main ways you can do this. You can do it through aerobic respiration, you can do it through anaerobic respiration, and then you can do it through the phosphocreatine pathway. Let's talk first about the aerobic respiration pathway. So the aerobic respiration pathway has three different steps. There's glycolysis, the Krebs cycle or the citric acid cycle, and then there's oxidative phosphorylation or the electron transport chain. And at each of these different steps in aerobic respiration, you're regenerating different amounts of ATP. So for instance, in glycolysis, which converts glucose to 2 molecules of pyruvate, you produce a net of 2 ATP from ADP. So you converted 2 ADP to ATP. You also produce 2 NADH, which are important for the electron transport chain. So just hold that in the back of your mind. You also made 2 NADH in glycolysis. And then in the Krebs cycle, which remember, for each pyruvate, you get 1 turn of the Krebs cycle. So you're getting 2 turns of the Krebs cycle per pyruvate. What you're doing in this process is you're first taking pyruvate, and via the pyruvate dehydrogenase complex, you're converting these 2 pyruvate molecules to 2 acetyl-CoAs, and this process produces 2 NADHs, and then you're feeding those pyruvates into the Krebs cycle pathway, where eventually you end up with oxaloacetate, and then the cycle starts again. So as this acetyl-CoA is converted to oxaloacetate, per glucose, you're producing 2 GTP, which I'm going to say that's equivalent to ATP, right? You're regenerating GTP from GDP, which is just guanosine triphosphate. So it's a different nucleotide. You're producing 2 of those. You're producing 6 NADHs. You're producing 2 FADH2s. And again, those 6 NADHs and 2 FADH2s are important for the electron transport chain, as I will mention next. Let me quickly summarize that because that was a lot of numbers coming in at you pretty hot. So in the intermediate step between glycolysis and the Krebs cycle, you're converting this pyruvate to acetyl-CoA. That's producing 2-NADH because you're converting 2 pyruvates to 2 acetyl-CoAs. And then once you feed those 2 acetyl-CoAs, which equal 2 turns of the Krebs cycle, you produce 2 GTP, 6 NADH, and 2 FADH2. Now, at the electron transport chain, what's going on is these electron carriers are going to this electron transport chain, donating off the electrons there. You're pumping hydrogen ions from the mitochondrial matrix through the membrane and into the intermembrane space in the mitochondria. That's creating a hydrogen ion gradient, which then turns this ATP synthase, which then converts a DP to ATP. Now, to determine the number of ADP that are converted to ATP at this electron transport chain, you need to know a few conversion factors for these electron carrier molecules. So for NADH, 1 NADH is equal to 2.5 ATP, and then 1 FADH2 is equal to 1.5 ATP. So overall, if you take the 2 ATP that were produced during glycolysis, the 2 GTP, or we'll just say ATP, that were produced during the Krebs cycle, and now you add that to the ATP that's generated at the electron transport chain from these NADH molecules and FADH2 molecules, you'll see that per glucose you get about 32 ATP from aerobic respiration. So before I go on to anaerobic respiration and I talk about how many ATP are generated there, I quickly want to mention a little bit about ATP synthase because it's a pretty sweet little protein. So this is a membrane protein that is in the mitochondria of cells, and it's part of the electron transport chain, as I said. And this is the part of the electron transport chain that uses that hydrogen ion concentration gradient that's created by the electron transport chain to actually drive the production of ATP from ADP. And how it does this is almost unbelievable in the way this happens. So let me describe this. So the ATP synthase is broken up into 2 portions. There's the F1 portion and the F0 portion, or F0 portion. The F0 portion is the membrane spanning portion. That's the part that's actually embedded in the mitochondrial membrane. And then the F1 portion is the portion of this protein that actually has this ATP synthesis activity. Now, this F1 portion has 3 active sites, and these 3 active sites are where 3 different processes take place. And this is where I like to imagine this F1 is like a merry-go-round with 3 distinct portions, like a pie chart that's cut exactly into 3 equal pieces. And these 3 equal pieces correspond to the 3 different active sites. And these active sites are called the L site, the T site, and the O site. Shout out ovaries on that last one. So I'll go through what exactly is going on at each of these active sites. So at the L site, which is also called the loose site, ADP and an inorganic phosphate, just a phosphate group, diffuse into the active site and reversibly bind. And at this point, the F1 rotates like a merry-go-round. And this rotation is driven by that proton gradient that has been established. And what this causes then is it causes this L site to have a conformational change. And in this conformational change, it actually gets into more of a tighter conformation. And at this point, it's going to be called the T site, or the tight site. And in this tight site, ATP forms spontaneously from this bound ADP and the phosphate group. And this reaction is catalyzed in this active site, specifically in this new T conformation. And essentially what I imagine is that I imagine this rotating and essentially what I imagine is I imagine that you have this ADP and this phosphate group, and they're bound to the L site, and then it rotates and has that conformation change where it becomes a lot tighter. And what that's doing is that's kind of pressing together the ADP and the phosphate group, and it's like they form the ATP because they're just pressed together so tightly. Hence the tight site, or the T site. And so after this ATP forms, again, The F1 rotates because of this proton gradient, and the active site again changes conformation. This time it changes to an open conformation and is hence called the O site or the open site. And at the open site, the ATP is released and it diffuses away from the active site. And then after this, the cycle is repeated. So the F1 rotates again, and now you're back to the L site. So that's aerobic metabolism, and you produce approximately 32 ATP per glucose molecule. Now for anaerobic metabolism, you are only going through the glycolysis part of this pathway. And if you remember back to one of the podcasts I did, I talked about how you generate lactate during this process, which helps you regenerate NAD+. Now, as I said, during the glycolysis part of the pathway, you're only producing a net of 2 ATP. So for anaerobic metabolism, you're only regenerating 2 ATP. That's it. The last pathway I want to talk about here in terms of ATP regeneration is the Phosphocreatine Pathway, which is an important pathway. So creatine phosphate is an amino acid with a phosphate group attached to it. And it's at highest concentrations within the muscles. So it's mostly found within the muscles. And what it's able to do is it's able to actually transfer this phosphate group from the amino acid to ADP in order to make ATP. Now, the enzyme that catalyzes this reaction is called creatine kinase. And what it does is it catalyzes the transfer of a phosphate group from creatine phosphate to ADP in order to create ATP and creatine. Now, this system is meant for more of like an energy burst than it is for long-term energy like, carbohydrates and glucose are. According to a paper I was reading by Berg et al., they said that this creatine phosphate system can, quote, power intense muscle contraction for 5 to 6 seconds, end quote. Another way to think about it is it kind of acts as an energy buffer system. So another paper I was reading, and this is from a textbook, nutritional biochemistry textbook, and it says that, quote, creatine phosphate helps maintain a constant concentration of ATP in muscle cells during sudden bursts of exercise that would otherwise deplete ATP concentrations inside the cell, end quote. I kind of like to think about this as like sprinting in a video game. Typically you have like a little bar on the side that is how much sprint you have, and then when you sprint, you deplete that sprint all the way down, and then you got to walk for a little bit, and you got to kind of walk until that sprint bar is recharged, and then you can sprint again. So in the same way, creatine phosphate exists so that you can quickly transfer this phosphate group from the creatine phosphate to ADP in order to regenerate ATP, but there's a limited source of this creatine phosphate, and after it's depleted, you can no longer use that creatine phosphate, at which point then you got to wait for it to regenerate, then you can use it again, and it only lasts for, as I said earlier, about 5 or 6 seconds, according to the paper I read. And the last thing I want to mention here about ATP regeneration goes back to aerobic metabolism. I want to make it clear that glucose breakdown in glycolysis and the Krebs cycle is not the only source of these high-energy electron carrier molecules, the NADH and the FADH2. NADH, FADH2, and acetyl-CoA can all be produced via the breakdown of fatty acids during what's known as beta oxidation. Now, during each round of beta oxidation, the fatty acid molecule is shortened by 2 carbons. And each of these rounds produces 1 NADH, which is worth 2.5 ATP, 1 FADH2, which is worth about 1.5 ATP, and 1 acetyl-CoA, which is worth about 28 to 30 ATP. So fatty acid oxidation indirectly produces a shit ton of ATP. For example, take a molecule of palmate, which is C16H32O2, which is one of the most famous fatty acids. It yields about 130 ATP. That's a lot of energy. So all this is to say that fatty acid breakdown can produce NADH and FADH2, which can be fed directly to the electron transport chain, and they can produce acetyl-CoA, which can be fed into the citric acid cycle. And all of those processes can produce ATP, or I should say regenerate ATP. Another group of molecules that we take a lot of in are amino acids. And we know there are a subset of amino acids that are called glucogenic amino acids. And it's all the amino acids except for leucine and lysine. And they can be converted to glucose through gluconeogenesis. And then this glucose can obviously be fed into the anaerobic or aerobic pathways that I just talked about. So, in a way, amino acids can contribute to the regeneration of ATP, although they do it indirectly, as is the case with fatty acids. I think the big takeaway from this part of the podcast is that aerobic respiration regenerates about 32 ATP per glucose, whereas anaerobic respiration produces, or I should say regenerates, about 2 ATP per glucose. On the other hand, the creatine phosphate pathway is this quick pathway that we can use to regenerate ATP in our muscles during these short bursts of exercise. All right, so those are the 3 quote-unquote regeneration pathways for ATP. I want to get into quickly the biosynthetic pathways for ATP. So as I said, ATP is a nucleotide, so the nucleotide biosynthetic pathway is technically kind of the ATP biosynthesis pathway, even though my understanding is that you're making adenosine monophosphate first, and then you're adding on those other phosphate groups later. So the pentose phosphate pathway produces ribose 5-phosphate, which is the precursor for nucleotides. So essentially, this ribose 5-phosphate is fed into different biosynthesis pathways for the purines and the pyrimidines in order to produce these different nucleotides. And that is about as detailed as I'm going to get into the pentose phosphate pathway. I don't know how high yield that is for the MCAT, so I'm going to kind of skip over going into great detail there. I kind of covered it in one of the metabolism podcasts, so if you want to go check it out there, you can. But just understand that the biosynthetic pathway of ATP is basically the nucleotide biosynthesis pathway. And this is going to produce from scratch new adenosine nucleotides, which is distinct from just regenerating ATP molecules from ADP.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
All right, so the next thing I want to go into here is the thermodynamics behind ATP. Let's start by talking about why ATP is such a high energy molecule. So the energy that ATP holds is in its phosphate-phosphate bond, specifically between the gamma phosphate and the beta phosphate groups. And this bond is called a phosphoanhydride bond. And the reason this bond holds all this energy is because it's really hard to form this bond. You got to put a lot of energy in to actually form this bond. And that's because these phosphate groups are negatively charged, right? A phosphate group alone is PO4 3-. It's got these negative oxygens on it. And so when you take these negatively charged phosphate groups and you pack 3 of them right next to each other on the same molecule, these phosphate groups just want to get the hell away from each other, but they can't because they're bonded together. So those bonds have to be pretty freaking strong for all of those to be held in and to counteract the repulsion forces that these charged phosphate groups feel towards one another. Now, once this terminal phosphoanhydride bond is broken, and that's just going to be the bond between, again, the gamma and the beta phosphate groups. Once that's broken, you produce an ADP and 1 phosphate group. And this ADP is a lot more stable than ATP. And one of the reasons this is the case is because there are multiple resonance structures for ADP, whereas there are none for ATP. And we know in general a molecule with multiple resonance structures is more stable than one without resonance structures. And resonance in general shows that electrons are capable of moving around, and therefore their charge is a bit more delocalized. And this helps spread this negative charge over the whole molecule and decreases some of the repulsive forces between these two remaining phosphate groups. So this is all to say that ATP is a highly energetic, somewhat unstable molecule. And there's a lot of energy stored in this group, in this bond between phosphate groups called the phosphoanhydride bond. All right, so let's get more into the thermodynamics of ATP. So ATP undergoes hydrolysis in order to be converted to ADP. And a hydrolysis reaction is just one where a water molecule is a reactant and you're breaking some bonds. So if you were to look at the full hydrolysis reaction of ATP, it would look like ATP plus water breaks down to ADP plus a phosphate group plus a hydronium ion, which is H3O+. And this reaction has a change in enthalpy, or a delta H, of about negative 65 kilojoules per mole. So that tells us that this reaction releases heat as a byproduct, and therefore is called an exothermic reaction. And this is one of the reasons why, as you begin to exercise and use more ATP in your muscles, your body temperature begins to rise. The change in Gibbs free energy, or the delta G for this reaction, is between negative 28 and negative 34 kilojoules per mole. And since this delta G is negative, this is an exergonic reaction, which is distinct from exothermic. Exothermic talks about enthalpy, whereas exergonic talks about Gibbs free energy. So it's an exergonic reaction, and in other words, it is a spontaneous reaction. So just to quickly summarize that, the change in enthalpy of this reaction is negative 65 kilojoules per mole, which tells us that it is an exothermic reaction and it releases heat. And then the delta G is about negative 30 kilojoules per mole, and that tells us that this is a highly exergonic reaction, or that it's spontaneous. In terms of the mechanism for this ATP hydrolysis reaction, it's actually a pretty simple mechanism, and I've broken it down into 3 separate steps. And I will note that if you want to go see this, there's a great Khan Academy video where this is all drawn out, so I recommend that if you want to see this in action. So in the first step, there's a water molecule floating by this ATP. What happens is the oxygen of this water molecule attacks the phosphorus of the gamma phosphate group. In step 2, which is occurring at the same time, the phosphoanhydride bond breaks, which again is this phosphorus oxygen bond between the gamma phosphate group and the beta phosphate group. So that bond breaks, and the electrons of that bond are given to the oxygen of the beta phosphate group. Now, in the final step of this mechanism, another water molecule comes along and it actually picks up one of the hydrogen from the original water that attacked that phosphorus group, right? Because when water attacks something, it's obviously got 2 hydrogens. Now it attacks something, it forms a bond, and now it's attached to 3 different things. So it has a positive 1 charge. In order to get rid of that positive charge, it's going to give away one of those hydrogens pretty easily. So a water molecule comes along and takes that, and now that water molecule is a hydronium ion, which is H3O+. And so overall in this reaction, you're creating a ADP molecule, a phosphate group, and then a hydronium ion, or H3O+. Now, when I talk about the delta H, or the enthalpy, and how that's a negative 65 kilojoules per mole, that means that this reaction is releasing energy. So one thing you might say, or one thing you might be thinking is, but wait, I thought that breaking bonds requires energy, right? We're breaking this bond between these phosphate groups. Doesn't that require energy? Why is this reaction releasing energy? In that line of thinking, it is somewhat true, right? It always is going to take energy to break bonds. It always does. But you're not thinking about the whole reaction, right? So the whole reaction is what I just explained, right? You're forming a bond with water, and you're breaking this phosphoanhydride bond. So what's happening overall is essentially that it takes only a little bit of energy to break that phosphoanhydride bond because it's so unstable, but then forming the water phosphorus bond releases a lot of energy. So then overall you're releasing energy even though you're breaking a bond and forming a bond. Hence you get this negative 65 kilojoules per mole for the enthalpy change. So what I want you to take away here is that yes, breaking bonds always requires energy, and yes, forming bonds always releases energy, but typically this isn't happening in a vacuum, right? You're forming bonds, you're breaking bonds, and then the overall enthalpy change is going to be, it's going to factor in both the bonds being formed and the bonds being broken. So just keep that in mind when we're talking about ATP hydrolysis and that reaction releasing a shit ton of energy. All right, the last thing I want to talk about in this section of the podcast is how ATP hydrolysis can be paired with other reactions in order to drive those reactions. So there are many biological reactions that are non-spontaneous. In other words, they have a positive change in Gibbs free energy for the reaction, but they can be paired with ATP hydrolysis to make them spontaneous. So take, for instance, peptide bond formation, which is an essential reaction, obviously, for producing polypeptides. If you and I were unable to form peptide bonds, then we would just be like a puddle of amino acids. You couldn't form these long chains, and I don't know what that would look like, but I'm guessing you'd be a puddle. So peptide bond formation, in terms of its change in Gibbs free energy, is positive 16 kilojoules per mole. Again, that's non-spontaneous. And then ATP hydrolysis, as I said, was around negative 30 kilojoules per mole for its change in Gibbs free energy. So if we pair these reactions together, add negative 30 to 16, it's about negative 14, which means that if we pair these together, it's going to be spontaneous. Now, how these reactions are paired together is a bit more complex, and I think that changes depending upon what reaction we're talking about. Like, it's not the same for peptide bond synthesis as it would be for nucleotide synthesis or, other things. So I'm going to go through a little bit of the actual mechanism for peptide bond synthesis and how that's paired with ATP hydrolysis. It's really complex, and I'm giving like a grossly simplified view of this mechanism. But what I want you to understand is that it's not like ATP is just being hydrolyzed and then that's on its own, and then ATP is just like, here you go, here's the energy, and just like hands over Gibbs free energy. Because Gibbs free energy is not like a tangible substance. It's just like this. Gibbs free energy is made up, honestly. It's made up. It's just this calculation we do. I don't even know what it is. It's made up. ATP can't hand over Gibbs free energy. So here's this grossly simplified mechanism. Step 1. Amino acid number 1 attacks the alpha phosphate of an ATP molecule. At this point, the beta and gamma phosphate groups leave. So now you have amino acid 1 attached to the alpha phosphate of essentially an AMP molecule, or an adenosine monophosphate molecule. Step number 2. The nitrogen of amino acid number 2 attacks the carbonyl carbon of amino acid number 1 that is in this AMP amino acid compound. And now at this point, the phosphoanhydride bond that had formed between amino acid 1 and this adenosine monophosphate molecule breaks. And in step 3, you have the dipeptide that has been formed. Again, the point here is that ATP doesn't just give its energy to amino acids to drive peptide bond formation. Rather, ATP is an active participant in the mechanism of peptide bond formation. All right, in this last segment of the podcast, I want to go over some of the functions of ATP. So I'll talk about the general function, which I'm sure you have a pretty good handle on, and then I'll talk about 3 specific cases. I'll talk about the sodium-potassium ATPase, or the sodium-potassium pump. I'll talk about protein kinases and protein phosphorylation, and then lastly, I'll talk about the ATP binding cassette transporter protein. So in general, ATP functions as an energy carrier. As I said, it's paired with many biological reactions, including DNA, RNA synthesis, protein synthesis, and it drives them in order to make them spontaneous. So that's the general case. The first specific case I want to talk about here is the sodium-potassium pump, or the sodium-potassium ATPase. So this is a membrane transporter that is very important for maintaining sodium and potassium intracellular concentrations, which helps maintain cells' resting membrane potential. Obviously, this is particularly important for neurons. However, there's many cell types that express this sodium-potassium pump, at least to some degree, not just neurons. And I think it's important to understand the mechanism here of how this pump operates. So the mechanism is as follows. First, the pump binds 3 intracellular sodium ions. Then it binds ATP. Then the ATP is hydrolyzed, leading to phosphorylation of the pump at an aspartate residue. At this point, the ADP that is left over is released and that floats off. And what this phosphorylation at this aspartate residue does is it actually causes a conformational change in the pump. This conformational change in the pump exposes the sodium ions to the outside of the cell. And I like to imagine this conformational change as like changing from a V to an upside down V. So like take your hands and make a V with your hands, and then the conformational change would be making an upside down V. In other words, like your palms would be touching to make the V. Now your fingertips are touching to make an upside down V. Now, once this pump is phosphorylated, as I said, it changes in conformation, but it also has a lower affinity for the sodium ions. So because of that, after this change takes place, the sodium ions are then released to the outside of the cell. Now, once that occurs, then the pump will bind 2 extracellular sodium ions, and this causes the dephosphorylation of the pump. Essentially, the phosphate group that is bound to the aspartate residue disassociates, and that causes it to revert to the previous conformational state. And now this unphosphorylated form of the pump has lower affinity for the potassium ions, and so those potassium ions now just diffuse into the cell. Now you start the cycle over where 3 intracellular sodium ions bind to the pump. And so in summary, the sodium-potassium pump moves 3 intracellular sodium to the outside of the cell and transports 2 extracellular potassium to the inside of the cell. And this is a process that is energy dependent and requires ATP. And then ATP phosphorylates an aspartate residue that is on the sodium-potassium pump. And an interesting application of a drug that actually blocks the sodium-potassium pump is medication that's given for people with heart failure. So heart failure is a disease where your heart fails to pump enough blood to all the different tissues in the body. And so there's a specific class of inhibitors called glycosides that bind to the extracellular part of the pump, i.e. the part of the pump that binds the potassium when it's in its phosphorylated state. And what this binding does is it actually inhibits the pump from functioning. It can't move sodium and potassium outside and inside of the cell. And what this causes is the sodium to start building up in the cell, which then inhibits a calcium transporter in the cell, which then causes calcium in the cell to build up. And as we know, calcium has a function in muscle contraction, and this buildup of calcium actually causes the smooth vesicular muscles to contract with a greater force, which then means you get better pumping, which means that you get less heart failure, right? You're able to get the blood around to different parts of the body better. So the next thing I want to talk about here is protein phosphorylation. And this is a type of post-translational modification, i.e. it happens after translation. And an enzyme called a protein kinase actually catalyzes the transfer of a phosphate group from ATP to a hydroxyl group of an amino acid residue in order to phosphorylate a protein. So let me just give a quick example. So say you have tyrosine, at position 182 on a protein, and it's going to get phosphorylated to activate this protein. Well, what happens then is you have a protein kinase come by, and it's got an ATP, and it takes the gamma phosphate group, and it transfers it from the ATP to the hydroxyl group on the tyrosine, and boom, the protein is phosphorylated. And so what this means is that for the most part, only amino acids that contain a hydroxyl group can be phosphorylated. So the most common of these are going to be serine, tyrosine, and threonine. Histidine is another one that can be phosphorylated, and it's actually phosphorylated at a nitrogen, not actually at a hydroxyl group. But those are the four I would remember: serine, tyrosine, threonine, and histidine. Those are the four most commonly phosphorylated amino acid residues. The less common ones are the other ones that contain hydroxyl groups. That's going to be aspartate and glutamate. In a textbook called Basic Neurochemistry, Molecular, Cellular, and Medical Aspects, 6th edition talks about why phosphorylating a protein changes its activity. Quote, because phosphate groups are highly negatively charged, phosphorylation of a protein alters its charge, which can then alter the conformation of the protein and ultimately alter its functional activity, end quote. And lastly, I'll mention too that it's not like every single tyrosine residue on a protein is capable of being phosphorylated. Typically, it's very specific residues that get phosphorylated, right? They've got to be exposed to the solution that the protein is in, right? In other words, the protein kinase that catalyzes this reaction must be able to actually reach the residue in order for it to get phosphorylated, right? It can't be, the residue can't be buried in the middle of the protein. That's never going to get phosphorylated. I'd say the big takeaway here is remember the four most commonly phosphorylated amino acid residues. That's going to be serine, tyrosine, threonine, and histidine. And the phosphate group that is phosphorylating the protein comes from an ATP, comes specifically from the gamma phosphate group. All right, the last thing I want to talk about in this podcast is a class of proteins called ATP binding cassette transporter proteins. So these are a very broad class of membrane proteins, and what they do is they function to pump molecules against their concentration gradient, which, as you probably know, is an energy-dependent process. So these require ATP to function. And these ATP binding cassette transporters, or ABC transporters, are characterized by the presence of two domains. The first being the nucleotide binding domain, which it has two of, and the transmembrane domain, which it also has two of. So overall, you can imagine it having two nucleotide binding domains and two transmembrane domains. And how it functions essentially at the most basic level is that ATP hydrolysis at the nucleotide binding domain drives a conformational change in the transmembrane domain, resulting in unidirectional transport of some molecule across the lipid bilayer. So that's another important point, is this only transports a molecule in one direction across the membrane. It's not like the ATP it's not like the sodium-potassium pump or the sodium-potassium ATPase where you're transporting sodiums out of the cell and potassium into the cell. It doesn't work that way. It's only going one direction. And a good example of one of these ABC transporters is a protein called ABCA1. And it's an important protein for cholesterol transport, and specifically reverse transport of cholesterol. If you're not familiar with reverse transport of cholesterol, essentially that's where HDL, which is high-density lipoprotein, which is the quote-unquote quote healthy cholesterol, takes cholesterol from the periphery. This is cells, at different parts in the body, and it takes that cholesterol from those cells and it moves it back to the liver. And from the liver, it can either then be passed on to LDL molecules, which will then take it back to the periphery if you need it in the cells, or it can be excreted through the gallbladder. So reverse transport, again, just moving it from the cells in the periphery back to the liver. And so this ABCA1 membrane protein is in the membrane of a cell, and what it does then is it transfers the cholesterol from inside the cell to the HDL particle, which binds at the surface of the cell. And this process requires energy because you're taking cholesterol from the cell where it's at a lower concentration to these little HDL particles where the cholesterol concentration is higher. So the takeaway here is that an ATP binding cassette transporter protein just transports different molecules against their concentration gradient unidirectionally with the help of ATP. All right, so the last thing I want to say here is just answer a question that I know is in your head right now, and that is, why don't we just eat ATP? Like, why aren't we just taking ATP supplements or injecting ourselves with ATP instead of eating food, right? If this is the source of energy we get from glucose, why aren't we just eating ATP? I have heard of NAD IVs, and it got me kind of thinking, like, is there such thing as an ATP IV? And if so, are people doing it? Are there ATP pills? If so, are people doing that? And so the first thing here is that after doing a little bit of research, find out that ATP oral supplements are not bioavailable. And there is strong evidence for this conclusion in a 2012 randomized placebo-controlled trial in healthy humans. So essentially what's going on is that if you take an ATP pill, all you're going to do is pee out all of that ATP. None of it is actually getting absorbed. Now, what about intravenous ATP? Well, as you know, ATP is a charged molecule, and so charged molecules have trouble making it through cell membranes in appreciable amounts without some transporter. Now, there is such a transporter that is produced in some cell types, but this is mostly only produced in erythrocytes, which are also called red blood cells. So my understanding of why we don't take ATP supplements goes like this. So oral supplementation of ATP is bad because you just urinate it all out. IV administration of ATP is okay. I mean, the ATP will make it into erythrocytes, and that's because of the expression of a specific receptor transporter that brings that ATP in, but it has a hard time making it into other cells in the body. So when you take that IV dose of ATP, it's not like it's going to make it into all the muscle cells, and it's going to help you. It's going to help you with the things you care most about, like muscle contraction. So this is all to say that next time you're thinking about taking ATP supplements, just eat a banana. Thanks for listening to this episode of MCAT Basics, put out by Med School Coach. As always, if you like what we do, please give us a review on iTunes. Helps us in the rankings, helps us reach more people, and good luck as you continue to study for the MCAT.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.